So I believe a couple of you are looking at secondary and tertiary structures within DNA, but I don't think anybody's talking about quaternary structure. So one of the things we'll end up finding is that, as will probably be discussed with the secondary and tertiary structure, is that your DNA will coil, and it can supercoil. As it supercoils, think of, this may be a bad, well, you guys are all old enough, maybe. You know the phones, like actual phones that weren't wireless, that had the cable on them, that little coil? And that coil would ever so often get kind of knotted up because you'd pick up the phone and you'd twist it, okay, and you could supercoil that cord, and it would coil up on itself. DNA does the same thing. Okay? You can also uncoil it. Okay? So depending on what stage you're at within your DNA storage or analysis mechanisms, it'll either be super wound or it'll be super unwound, depending on what you're trying to do. Okay? The more wound up it becomes, what do we think? Smaller area, it's probably not going to replicate. We've compacted it down so much, it makes it much more difficult to get access to the genetic material contained within it, which I guess starts to reveal some of the secondary structure. Okay?
So why does it do that?
Protection. Okay? Your genetic material is your, it's your key to make everything that you potentially need. If that is then permanently exposed, that then means it has a permanent chance to react. If it has a permanent chance to react, it can then get destroyed. Okay? So it is a protection mechanism to wind it up and keep it contained and protected. We then need to be able to unwind it to get access to it when we need to get access to it, say to build new things, okay, new proteins, because our old proteins got degraded, or to replicate the cell. Okay?
So they know the chemical, like, so I know, like, on the outside, you know how you have negative charges on them, that they say that it attracts, then you're going to get positive charges to attract it. So is it those proteins that are coming in that are causing it to do the coiling, or is it? So
there are specific proteins involved in getting it to coil, yes. And their names are helicases. Again, kind of giving tips as to what's happening with our secondary structure, but they will help wind or unwind depending on the need at a particular instant. And that need is going to be represented by subtle chemical changes within the system. Okay. As it supercoils, it will start to form loops. Okay. Well, we want to protect those loops as well. I mean, if we took something fragile, because what we're looking at here is kind of like dry spaghetti. Okay. We can wind it, and eventually it will loop back on itself, but now you have a highly stressed system. If you then compressed that loop, what happens? It'll break. So we want to shove something in between there to say, okay, now when it gets compressed, it can't break. That's our histones. So our histones will sit inside those supercoils to kind of help protect, potentially, the DNA, and they could potentially also be used to help kind of uncoil and expose the DNA around them. Okay? That exact mechanism isn't perfectly identified yet. Okay, so it is a super active area of research to figure out what exactly the histones are doing, because it makes sense for them to potentially be protecting it, but just as easily you could then kind of further expose the DNA specifically around the histones. So the histones could help open the DNA at those individual locations so that we could get access to the material.
Could the histone also help stabilize it?
Yes.
Whatever
well, so that's one of the theories.
Hold
yep.
on to all the
Yep, so it can hold on to all those pieces and keep them all kind of protected and wound up. Okay? It could also provide binding sites for another enzyme to come in. The enzyme could bind to the histone, and since the histone's bound to the DNA, it can now have better access to the DNA.
It would help to stabilize the enzyme so that it doesn't have to rush into the DNA.
Entirely, yeah. So, variety of different mechanisms being discussed there. It's kind of hard to nail down exactly what's going on with it yet. They're still working on it. Okay? But it is kind of neat. We do know these structures exist. They can be seen through microscopes, and that's where we get chromatin. Okay? Chromatin is your DNA strand then wrapped with your histones. Okay? And you can see that if you do your, mitosis, all that fun stuff, you can see DNA start to replicate and split, and it's because those have now become super densed. Super densed. Is densed a verb? okay, densified. It's not getting any better, around our histones, which makes it more visible because they're now compacted into a tinier area. Okay. So kind of neat. Other parts of our genetic material, we end up having ribonucleic acid. Some of the big differences between RNA and DNA.
RNA is found both in the nucleus and in the cytoplasm.
Okay. RNA is found in both locations. Okay. And you mentioned nucleus, but do all cells have a nucleus?
No.
No. Okay.
Prokaryotes
Prokaryotes don't. So in eukaryotes, yes, the DNA is found only in the nucleus. RNA is found in both locations, both nucleus and outside the nucleus. Even in prokaryotes, DNA is still kind of centrally located. It's just not behind a membrane. Okay? So the RNA can pretty much go wherever it wants, which is one of the reasons why RNA is considered a potential for the starting of all life, because RNA holds genetic information because it's made of nucleotides, and it has the potential to do enzymatic activity. Okay? So it has the best of both the DNA and the protein worlds. So there is a high probability, or a potential probability, that RNA is our origin of life that was then further specialized into DNA to make specifically genetic information, and further specialized into proteins to make our enzymatic activity. Okay? So it's kind of a nice intermediary. So that's a big difference. Where else is there a difference?
The sugar?
What do you mean the sugar? You want to elaborate?
I was just going to say that the DNA stores the information, but doesn't really do anything beyond that. But
So the individual roles of DNA versus RNA.
Actually transfers the information to proteins. And to
Yep. DNA is our permanent storage place. RNA takes that storage and can then use it. Okay, so it's still holding genetic information, but it then uses that to build other things. Okay, so that comes back to their roles. What else is a big difference between DNA and RNA?
It's ribose and deoxyribose.
Yeah. The sugars are both ribose sugars, but DNA is the deoxy form. So at position 2, we have hydrogens. The other positions all have our OHs. So there's our deoxy form. Our RNA, I think I did that correctly, has the oxy form. Okay. Again, at position 2. I think I have that OH in the proper spot. I'm not positive though. Okay. Why does that make a difference? We did talk about that, I think, on Tuesday. Oh, Mike, you shook your head yes that we talked about it, so why?
Yeah, we did. Because it's, by having the other oxygen, it's more electronegative, so it's more reactive.
Yeah, RNA is going to be much more reactive because at that position 2, we have that alcohol. Oxygen being more electronegative generates a polarity within the ribose sugar. That position can now potentially be altered and reacted. Whereas in DNA, we lose that reactivity, which goes back again to their individual roles. DNA, we want to be incredibly stable. We don't want it to react. RNA, we do. Okay? Or we want the flexibility to be able to react. Okay? So we have some pretty significant structural differences there. Is there another structural difference? Okay. Going to our secondary structure, which we officially haven't talked about. DNA is double-stranded. RNA is single-stranded. I guess I could be writing some of these down. It might pop up. What else?
And then the bases, so for the purine, is it purine? It just has U instead of T. Instead of thymine.
So our purines versus our pyrimidines. Remember the little trick on remembering? Oh,
Yeah,
I remember the trick, but I don't remember which one's which, so that wasn't very helpful. So our purines are our larger structures, so those are our two-ring structure, and then the pyrimidines, because of the larger name, is the single ring, which I think was still a six-membered ring. Okay. Within the pyrimidines, which nucleobases do we have?
A, T, and U.
A, T, and U. And then in the purines, we have G and C. Wait, is that right?
Yep.
What is A bond to?
Oh, no, it's G.
No, yeah, I was going to say something's not right there. That messes up the spacing. So let's try that again. Pyrimidines were
T, U, and C.
okay, and the purines were then
G and A.
G and A. Okay. Oops, no, I did like that order I had. Okay. Where does DNA use these? Or which of those does DNA have access to?
A, G, C, and T.
DNA is A, G, C, and T. Okay. Where's U used? Just with our RNA. Okay, or primarily with our RNA. U we won't see in DNA, so there's another big difference there. Okay. RNA gets access to uracil. That's a U. Okay. So there's some of our primary differences between those that should help us out. Kind of general questions on deciding between those two. Kind of make sense? Let's see, do I provide a big list? So single-stranded uracil versus ribose. And then we've got some of our kind of other summaries that we already came up with. Okay. DNA is our permanent storer of information. RNA transfers that information and can manipulate and use it. Okay. Even though it still holds that information. Okay. Types of RNA. We are finding more types as history progresses, so you may see more than these, but these are the most common ones. The most common, actually, I guess I should be fair, the most common are those first three. So if you're looking at, say, standardized tests, you should absolutely know those first three. The last two, and then there's, I think, at least one more. I don't remember. It's code, but there's a couple others, I think. Okay. So those first three, super, super important. You need to know those for any kind of standardized test. You have tRNA, where the T stands for transfer. So its role in life is going to be transfer, one thing to another thing. Okay? So really what it's transferring are your amino acids. Okay? In the grand scheme of RNA molecules, it is relatively small. rRNA, the R stands for ribosomal. Okay? And what happens in the ribosome?
Translation.
Where the proteins are put.
Yes, it's ultimately our translation, okay, which involves all of these things, so we want to be a little bit more careful or a little more particular with that. The ribosomal RNA is the enzyme that puts it all together. Okay? So it is the one responsible for the putting the doing that final translation. Okay? Taking information from one piece of RNA and the information from another piece of RNA to synthesize proteins. So it is our enzymatic core, is our RNA. mRNA is messenger. It's the one that transfers information from the DNA out to the ribosome, okay, to our rRNA to go through and process. Okay? So our mRNA is kind of our genetic information. Okay? It's the thing that holds the code that we then have to be able to interpret. Okay? And that's what the rRNA does, is it interprets the code and then brings in the appropriate tRNA with the proper amino acid to then build a protein. Okay? So the three of those all work kind of in concert to help go through and process. Because RNA still has genetic information in it, it can directly interact with DNA. Okay? So there are a variety of different mechanisms that could potentially come up with different pieces of RNA, and that's where we run into some of these other ones. Okay? snRNA, okay, is for, I think, small nuclear RNA. Okay, so it is typically smaller than tRNA, I believe. Okay? It's used to process our mRNA before it leaves the nucleus. So when we do our first transcription from DNA to RNA, there's a bunch of extra stuff in there that we don't need, okay? Which I think one of you guys will be talking about, maybe. I don't think you, maybe you. You're going to be talking about that? Okay, I might, or that might actually be, that might be Jimmy, okay? So I might bring in a lecturer to give us some of that information, okay? So our mRNA needs to get processed a little bit. That's where our small nuclear RNA comes in handy. It can help with that processing and remove the junk that we don't need so that the mRNA is now functional and can go out and be processed appropriately. If it gets processed as is, there's a bunch of stuff that goes into it that we don't want. That screws up protein structure. Right? And then there's the siRNA. Again, the S is for small, okay, and this time the I is for interfering. So when we're looking at our gene expression, how we interpret DNA and how it ultimately gets translated out to proteins or RNA, different things need to bind to the DNA, primarily of which we need to be able to put in new nucleotides if we're going to do replication. Okay? So the siRNA will actually match certain codes within the DNA and interfere with that process. If it comes in and binds, well then our enzyme or our enzyme that's processing DNA will come in and be like, well, crap, I can't process anymore because something's in the way. Okay? So it interferes with the expression of particular genes because it's binding and preventing that process from continuing forwards. Okay?
So we just need to figure out how to do that in cancer genes to stop them from replicating.
It's not just that we need to figure that in cancer genes, we need to figure that out specifically in cancer genes. Okay? Because the issue with most treatments when you're looking at cancer is we do a great job of destroying DNA.
All DNA.
All DNA. It is not specific. So we end up killing everything that is fast growing. The hope is that our extremely fast growing cancer cells die off because they can't reproduce anymore. Okay? And that the rest of our body that is relatively slow growing lives. Okay? We do have some cells that grow really fast, like skin and hair. Okay? Both of those things produce at very high levels. Okay? Which then means when we go through cancer treatment, what happens? We get a lot of hair loss because we stop producing hair. Okay? Because that requires fast-growing cells. Okay? Kind of makes sense? At least when we tie back to cancer.
Is the interfering also what would make the messenger RNA stop copying what it needs to?
Mm-hmm. So yeah, that
So it's
interfering
actually
RNA.
with what's going on, so
Yep.
they can get whatever they need.
Yep. And it may be very important that we don't actually start processing things. So when we look at genetic information, it's all stored there, and it is constantly interacting with the appropriate proteins and enzymes to replicate or to translate, or transcript. Okay? That is constantly going on. Do we need it to be constantly going on? Sometimes, yes, but most of the time, no. We don't need multiple copies of things. So something needs to prevent that process from happening. That's where our siRNA comes into play. And the hope is that it's really just slowing down that replication process long enough that our replication only happens every so often, okay, or once in the lifetime of the cell. Okay. Same idea with our transcriptions and translations. Our tRNA, so it's the smallest of those three primary RNAs. I don't think it's smaller than the Si or the SN. I'm not SN? Yeah. I'm not positive on that. If we look at the number of residues, 73 to 94, right, is how many different nucleotides go in to make the tRNA. Right? You'll notice that we're also introducing kind of a new term, 25 kilodaltons. Dalton is just a form of mass. Okay? And I believe it is actually just as straightforward as grams per mole. Okay? Why do we not say grams per mole? Because that takes too long, and we want to be nice to Dalton. Okay? And we hate students, because now they have to remember something else. Okay? So if you see a really large kilodalton or a really large Dalton value, you're just saying that that's a really, really, really big thing. If you're saying a small one, it's a relatively small structure. Okay? When we look at our TNA, we've got a general kind of goofy yellow picture in the middle there. What that's trying to show is how the single strand, okay, and so our single strand is kind of that outside, folds up on itself. Okay? Remember, our nucleotides, as we'll learn with our secondary structure, interact with another strand of DNA. Okay? Well, in RNA, if it's single-stranded, we now have half of that molecule is exposed to potentially interact through hydrogen bonding or hydrophobic interactions with another strand. Okay? Or it can react with itself. Okay? And it can fold up on itself to generate these linkages in between. Those linkages or those ladders are just that kind of interaction between the single strand. So it just folds up to generate a new kind of structure, much the same way we would expect of our proteins. At one end of it, okay, we've got a little hanging section here of nucleotide information, and then typically at the other end of it, we have information where we can pick up an amino acid. Okay. Every amino acid that is used in tRNA has a specific code. Okay, so each amino acid has its own version of tRNA, okay, which means there has to be at least 20 tRNA molecules or types of tRNAs, so that they can each carry their own individual amino acid. Why do we need different ones? Why not just use the same tRNA for each amino acid?
Because then you wouldn't have the amino acids in the correct order.
What do you mean? Why would we not generate our proteins in the correct order?
Because if it was all the same one, then the hanging information off the end would all be the same.
We have to have a code that correlates to here's some nucleotide information and here's the protein or the amino acid. If that code is identical for every amino acid, we get every amino acid built or every protein built with a completely random and arbitrary amino acid chain linkage or primary structure. Okay? So we need very particular codes coming out of that. That's where we end up with the codons, okay, that code for particular amino acids. So we need a tRNA to be able to recognize that code and to associate that correctly with the proper amino acid. Okay?
So you said it picks it up at that 2 end, but you said it carries amino acid at the 3' end.
Did I do it backwards? Oh, I did it backwards. Yeah. So our amino acid is up here.
Okay.
And at the bottom is our DNA code. I believe is how it codes. Okay. tRNA will need to be activated, so that's what it's referencing down at the bottom. tRNA nucleotidal transferase. Okay. So once tRNA is made, we actually have to activate it and say, okay, now go pick up an amino acid. Okay. That enzyme has to be able to activate it, okay, before it's useful tRNA. That enzyme will go through and add CCA to tRNA that is functional or correct. It'll add CCA to bad tRNA. So another enzyme will go through and be like, oh, what's this? Why is there a double repeat of the CCA? Okay. As soon as it recognizes that, it destroys the tRNA. It says, whoop, bad thing, kill it, because it's going to do something wrong. Okay. We then have all the pieces again that we could then rebuild our tRNA. So it's kind of a checking process. Okay. Our nucleotidal transferase processes the tRNA and says, yep, this is good to go out and do your job, or knock it off. Okay, I'm going to kill you.
Does the CCA get attached to the amino acid? end, or?
I think it gets attached to the amino acid end. Yeah. It's going to come down to how it can link in there. The easiest place to link is going to be off of your 3' end.
And all of them get those 3
Yes.
amino acids.
Nucleotides. CCA is your
Yeah,
nucleotide. Okay. Ribosomal RNA, 60 to 65% of it is found in ribosomes. Hence, they're called ribosomal RNA because that's where they're found. Okay, relatively straightforward. What we've got here is a kind of nifty picture of a ribosome from E. coli. And if you look really, really carefully, it's not super easy. It's a little bit easier, I think, to see up on the screen there. What does that kind of look like? Okay, this is really going to be appropriate for you, Leanna, because you've officially seen our secondary structure.
It's like it's traveling.
It's a helix. Okay. We actually have something on the inside of that. The RNA has actually been crystallized around so that we can see it actually processing a bit of a helix. Okay. Where are we going to get the helix from? Likely from our messenger RNA. Okay. When we look at the ribosome, or this particular ribosomal RNA, it gets subdivided into 2 categories, a 30S and a 50S. Okay, what do the S's stand for? I particularly find this annoying. Nope. This actually ties back to some of your studies, believe it or not. That S is the unit used to describe the density of the protein. So what someone went through and did was isolated ribosomal RNA and then did an ultracentrifugation process, which Robin will tell us a little bit about when he gets to his discussion. And those 2 pieces of our RNA, or those 2 things, end up being 2 pieces. That's why they're different colored there. And they settle out at different densities. Okay, because one part has a greater mass to volume ratio than the other, and we see them separate. Okay, so the S is just saying 30S means it has a lower density than 50S because 30 is lower than 50. That's it. Okay, so that's what that S is representing. If you look within the 30S, it turns out that you also have a 16S rRNA. So the 30S is actually composed of multiple other subunits. The largest of those is the 16S. There's officially 21 different proteins going into it. Okay? So depending on the level of the centrifugation and the accuracy that you can look at each of the individual pieces that make it up. When we look at the 50S version, that can break down into a 5S rRNA section, a 23S rRNA section, and then 34 other different proteins. And I guess I shouldn't say other, because that implies that they're the same thing. RNA versus proteins, different, right? RNA is your nucleotides, and proteins are amino acids. So when we look at the ribosomal RNA, the ribosome coming from E. coli is made up of lots of RNA and lots of proteins. So putting that all together, we are looking at a relatively massively sized complex to be able to go through and process our messenger RNA. Okay? Your replication. Sucks that Jimmy's not here, because then I could ask him if I should be stopping talking about this, because he might be talking about it. Messenger RNA, this is the one that codes for your information that the ribosome can then process. Right? So it's the one that carries the genetic information from your DNA out to the ribosomes for protein synthesis. Interestingly enough, the mRNA has an exceptionally short lifespan. Why would we want it to have a very short lifespan?
If it's making a specific enzyme, you may not want to keep making.
The point of the mRNA is to make something. Okay? I don't want to continue to make that. Okay? Once it's done its job, I want to get rid of it and take those pieces and put them back into the nucleus, because then we can then replicate or build other enzymes. So the mRNA tends to have a very short lifetime. As soon as it's spit out into our cytosol or our ribosome, it then gets processed very quickly, okay, and then very quickly degraded. Why might it be really quick to be degraded?
Good stuff. Need it.
Okay, we need it. Why else?
Bonds aren't very.
tRNA folded up on itself, rRNA folded up on itself, and had proteins come in.
Oh, messenger RNA is just a little.
The whole point of mRNA is to read it off as a strand, which means theoretically its lifetime, it's very elongated and exposed, meaning
It's gonna react.
there's a lot of reactive sites on that structure, which it needs to be read. If it folds up on itself, we can't read it. Okay, so it's now exceptionally exposed in its lifetime because of its role, which means anything present that's there to degrade, say, proteins or nucleotides so that we can then take those pieces back to be reused, is going to grab a hold of it very quickly and start to degrade it. Okay?
Is there a mechanism in mRNA that prevents it from folding in on itself?
That's an interesting question. The folding on itself ultimately comes back to how those individual sequences can potentially interact with each other. And what it may be is just that your tRNA and your rRNA are designed to fold back on themselves, so they have very specific folds that can keep them kind of self-contained.
Whatever sequences are, it just naturally.
Yep. And then in mRNA, it's probably not so much that something that its code prevents it from folding, it's that it was not designed to fold. Where our rRNA and our tRNA were designed to fold up on each other for their function. Okay? There may be a code within the mRNA that when it's first produced that the cell recognizes, hey, this is messenger RNA, so there might be a leader code on it saying this needs to get kept flat. So other enzymes, or potentially even our small nuclear proteins or some small interfering proteins,
Help it to stay.
may bind to it to prevent it from being, number one, decomposed, okay, and number two, to keep it relatively linear. But that I'm literally just making up. It just kind of makes sense to have something like that in the cell. It is synthesized from DNA starting at your 3' end, and it must be processed prior to leaving the nucleus. Okay, and that's where our small nuclear ribonucleic proteins, so that's the SNRP, SNRNP, okay, those are typically referred to as SNRPs, which are your small nuclear RNAs and proteins, okay, interacting with each other to go through and process and cleave out the stuff that we don't want. The bad stuff are known as introns, the good stuff known as exons. That sounds weird to me. Is that what you guys remember from biology?
I remember seeing the words, but I don't remember which was which though.
I mean, names don't always make sense, so that could very well be correct. That just seems weird to me. Right, and I don't remember. One of them's good, one of them's bad. Okay. Oh, there we go. There's the end of our kind of nucleotide information. And I wouldn't be surprised if I have a stupid question. Yep, there it is. On our exons and introns. So let's take a look at some of these. Which of the following groups is not found in nucleotides? They're all found in nucleotides. How could we change that question?
Change it to
No
nucleobase.
acid or something.
Or,
If we put in nucleobases, are they found in nucleotides?
no, you said found in nucleobases.
Oh, I see what you're saying. So instead of saying found in nucleotides,
Which
we-
ones are?
change that to nucleobases.
And then phosphates and sugars would be in those.
Phosphates and sugars are not in your nucleobase. When are sugars included? The nucleoside,
And then, yeah.
and then in our nucleotide, we have the phosphate added, and everything's in there. Okay. Yeah, I know that's kind of annoying, but just kind of have to deal with it. How could I change the answers?
You could put like,
Put an amino acid.
I could just say straight up an amino acid. That might be too easy, so I could jump in and say alanine. Okay. Why might I pick alanine?
It's close.
Close to adenine. Okay. So kind of be careful with that. Just make sure you're interpreting. The only things that go in are your nucleobases. Adenine, guanine, thiamine, cysteine, and potentially uracil. Because here it's not saying DNA or RNA. Okay. Which sequence of DNA should result in a higher DNA melting temperature? I really like this question. Though, crap, I can't ask it because it's based off of your material, Anna. So I can't actually ask that question. And the same deal with the one to the upper right. Both of those questions I haven't talked about. So both of those would be within the context of your material, Leanna.
Yeah, but I don't
So-
cover that, that's why I'm like, I don't know for the DNA melting temperature.
You might indirectly. Okay, so let's hold off on the DNA melting temperature for the moment, and let's move up to the other one. Which of the following statements is true only for DNA? Okay. So again, Leanna should be able to figure that one out. Mike and Robin, you're basing it off of your previous biology experience,
So
not
C.
from this class.
Oh, is true only for DNA, so that's not true, C is not true.
So C can't be true because that's true only for RNA.
And adenine was a purine.
Adenine-
No, it was a purine, no, it was,
I don't even remember which, but
it was.
adenine shows up in both DNA and RNA, so it's not true only for DNA. So C and D are wrong. The 3,5 phosphodiester link,
Oh, that's true.
that's true for both DNA and RNA. They're always linked through that same connection. So that really means that A is the only answer. Well, what's going on with that? That just seems a little bit weird. What we're saying with that is that when we look at DNA, we have equal amounts of G as we have C.
Because the double strand helix, and so you're going, okay.
So in DNA, it's a double-stranded helix,
In RNA.
okay, where G pairs with C.
Yeah,
So if we have a double strand, anytime I see G, what must be on the other strand?
C
C. So the relative ratio of G to C
will be.
will always be equal to each other. Whereas in RNA, because it's not double-stranded, there's no partnering on it. So in a single strand, it's possible that G is equal to C, but not always. Kind of makes sense? So it's a very kind of careful analysis of what is DNA. It's that double-stranded aspect. Mike, because I know you're doing MCAT kind of stuff, I would say this is a pretty legit question for the MCAT, okay? Because it's really pushing at what is happening within your DNA structure. So kind of watch out for that one. Higher sequence of DNA should result in a higher melting temperature. Okay, and this one becomes a little bit tricky. And sorry, Leanna, you're gonna get pushed here. Those are our pairs. That's not. Yeah, that's our pairs. Okay. How are those linked?
With hydrogen bonds.
How many hydrogen bonds between A and T?
I don't remember. I
Okay.
should know that.
Yeah, it's one. Well, kind of hard to say whether you should know that or not. Okay. A and T, and you should check me on this, have 2 hydrogen bonding interactions between them. C and G have.
Oh, because of the pyrimidines and the purines, like they're, okay.
It's how they link with each other. G and C end up with 3 hydrogen bonding interactions. A and T end up with only 2. So when you look at the double helix, and you will likely, if you don't already have a figure for this, Leanna, you need this in your double helix description, showing the hydrogen bonding interactions between those 2. Okay, between A and T and between G and C. And this becomes relevant for this question. If G and C have 3 hydrogen bonding interactions and A and T only have 2, what does that mean about the melting temperature? So your melting temperature is the energy it takes to split the 2 away from each other.
So the two are gonna break at lower temperatures than the three. It's gonna take more energy to break the three.
3 hydrogen bonding interactions is a higher energy interaction. It's going to require more heat. So the more G's and C's we see within a strand of DNA, the higher the melting temperature, which then takes us to B being our answer.
Two though.
Okay. This is a little bit shaky on an MCAT question, but it's possible you could see this.
I just did actually.
Oh, you did?
Practice question that was almost exactly the same thing.
Yeah. Okay, well then there you go.
They gave it like a long chain.
A big, massive chain, yeah.
Yeah.
I don't feel like writing that much. Which of the following is false for mRNA? God, I hate false questions.
It only contains introns,
Well, that kind of goes back to whichever one.
because the mRNA, you said that the exons were the jump.
So mRNA does contain introns. It also contains exons until it's been cleaved out of it. Once it's synthesized, it's mRNA. We then remove the bad stuff, okay? But that bad stuff was still in it. We then remove it to make it active mRNA.
Gotcha, so it's not activated until it leaves the.
It's not activated until it leaves the nucleus, and regardless, it still had introns and exons in it.
So it's D, it doesn't move
D.
amino acids.
Okay. Moving amino acids is a transfer process.
Transfer RNA.
That's transfer RNA. Okay. So this comes back to just knowing the definitions of what each of those types of nucleotides does. Sorry, not nucleotides. RNAs do. Which of the following are examples of RNA? Well, that's kind of a boring question. So all of them. The SI is your small interfering RNA. Okay. You should just kind of be aware of it. Okay. Kind of a boring question. Questions about that? Okay. The two starred ones, I personally do really, really like. They're going to be harder questions to ask. I might base them off of Leanna's presentation, and we've now talked about it, so we kind of have the context on it. So.
Hey, I thought I was supposed to come up with questions for my presentation, not you.
Okay, well, we could put it this way.
And I'm giving them to everyone.
Put it this way, you now have two questions that you could potentially ask. You do have to make sure your presentation covers it, but yes. Okay. If you've downloaded the slides, I apologize for this. I did a massive slide change. The lipids are like 10 or 20 or 30 slides later. So I'll give you a second to see if you can't track it down and find it. About within the lipid chapter, but there isn't a whole lot because a lot of it is just, this is a lipid. This is also a lipid. This is a lipid. You change this structure, guess what? It's a lipid. Okay. So short of just saying, here's a bunch of structures, you need to be able to name each of those lipids. That's kind of boring. So you won't see questions like that, but I should at least show you those structures. So lipids as a group are based off of their solubility. Okay. If we take a look at, well, I only gave you one example down there. So we can take a look at two. So we got examples of some lipids in the lower right-hand corner. Okay, your cholesterols, your testosterone, estradiol, and progesterone. Take a look at those, and if we take a look at our classic lipids, our fatty acids, what do those structures all have in common? I don't agree with that. Cholesterol is not linear. So your fatty acids are absolutely all linear, but cholesterol, testosterone, and all those are not linear. Your steroids are not linear.
They're predominantly carbon and hydrogen.
So we get a lot of carbon and hydrogen. So all those are carbon and hydrogens. Okay. All of those are carbons and hydrogens. Did I circle the whole structure?
No, it's the one end has
What did I leave off?
There's still
the end.
a few.
One end is not. That one end has a whole bunch of oxygens. So when we look at the solubility of our lipids, okay, one end is,
Soluble.
give me more,
Has oxygen.
soluble in
Water.
water. And the other end is hydrophobic, being insoluble in water. I was hoping you were going to go with hydrophobic instead of this insolubility thing, but we started with solubility, so I'm going to write it this way. So one end is hydrophilic, or soluble in water. The other end is hydrophobic, or insoluble in water. Okay? That is your standard classification for your lipids. Okay? Because all of those words were too many words, we invented a new term. That new term is amphipathic, meaning
Full soluble and insoluble, I don't know.
it does both. That's it. Think of amphibians. Okay, what are amphibians? They can live both on land and in water. They have the ability to go both directions. Okay, so that's what's happening with all of these. So you have some cyclic forms, and then you also have some straight chain forms. And then you might have jumped with the straight chain by looking at the fatty acids. There's a lot more straight chain or open chain forms than there are of the cyclic forms. Okay, at least as bulk classifications. Okay. So primary one we'll look at will be our fatty acids. So we've got a bunch of example fatty acids shown. Your textbook likes to go through and show these kind of three-dimensional models as well. The intent behind that is to show kind of their shape, and in particular, when it comes back to this fun concept of saturated versus unsaturated. Okay. Saturated means no double bonds. Okay. Unsaturated means double bonds. How is that going to change the overall structure or information about our
Double
fatty
bonds
acids?
kink.
The double bonds provide a kink within the structure.
Makes it easier for you to break down.
So if we looked at trying to stack, say, this straight chain form, those are going to fit together relatively nicely. But if I go through and put a kink in it, do they fit together quite as nicely? No. What does that mean happens to the interactions between individual chains?
It's not as strong because it's unsaturated. It's
Not as strong of an interaction, which then means the melting point should
lower.
lower. So the more unsaturation, the more unsaturation we have, the more liquid we should expect it to be. Okay? So if we go back and look at our saturated versus our unsaturated, okay, within our fatty acids, we tend to refer to fatty acids found in animals as fats, and those found in plants as oils. Same thing. They're both fatty acids, but what's the difference between them?
One's liquid.
One is liquid and one is solid. To become a liquid, what did we need?
Unsaturated.
We needed unsaturation. Which one's a liquid?
Oil.
The oils. Our plants tend to be all unsaturated, whereas our animals all tend to be saturated fats. So what's the difference between butter and margarine? Butter comes from
Cow.
cows, and margarine comes from
A test tube.
sort
It
of.
does, yeah.
Yeah, I know where you're going with that, but let's take it a step backwards from the test tube.
Like which-
Plants. It's usually the canola plant, I believe. I could be wrong on that one, but it is for sure coming from a plant. Okay, both of those are at least solid. Well, why is that the case?
Because they test tube canola.
So the canola stuff, what did they do?
They forced it to become saturated.
They are forcing it to become saturated by pumping hydrogen into it and catalytically converting it from the unsaturated form to the saturated form. Okay, that then increases the melting point, so we can take fats that are unhealthy, quote unquote, because nobody watching the video can see that, animal fats that are all nice and solid and easily spreadable via butter, and we can convert our plant fats that are healthy because they are liquids that we can then spread as well. Okay? And that's ultimately where margarine came from. It was that, well, if animal fats were bad, plant fats are good, so let's make spreadable good fats. Okay? That was the intent. Did we find out later that that was complete nonsense? Hell yeah. Okay?
It's so stupid.
But that's where the origin came from. Okay? So we're coming back to that idea and trying to figure out what's going on within it. One of the issues that comes out of this is that when we look at the unsaturated fats of our oils, okay, what type of unsaturation do we have? So if we look at, say, this double bond versus this double bond.
Cis
cis and trans. Cis meaning that they're on the same side, or the larger groups are on the same side of our double bond, versus trans meaning they're on opposite sides. When we look at our unsaturated oils, which do we typically have? We have cis. It's the trans fatty acids that are bad. Okay? Why are the trans fatty acids bad? Go back and look at your structures. What happens when you have trans fatty acids? You get larger kinks within your structure. Or sorry, not larger kinks. You get a straight chain.
It's easier to stack.
Okay? It is easier to stack. And the point of our cis is to kind of break that up and get a little bit more fluidity and mobility within it. So when we start with our cis canola oils and we go through to hydrogenate by adding hydrogen, we add hydrogen to some of the double bonds, not all of them. Unfortunately, we add enough energy into the system that we can convert between the cis and the trans forms. So the double bonds that were cis, that stayed around, that didn't get hydrogenated, become trans. And we end up generating trans fatty acids as opposed to the cis. And that messes with our whole system. Okay?
Is that it's harder for the body to recognize what it's trying, or
Body doesn't process it quite as well. Yeah. One of the things that I thought was interesting about this is our plant fatty acids were more liquid. Where do our fatty acids typically accumulate?
in the cell wall?
Cell walls, our membranes. Okay. Where would you expect to have more rigid cells? Plants or animals? Plants,
But
right?
plants have the cellulose.
If we look at plant cell walls, according to this, they are more fluid and mobile than animals, which makes no sense because plants don't move. So what else is going on with plants? They have a cell wall.
Cellulose.
Okay? That cell wall encapsulates it and provides the rigidity that the cell membrane does not. Okay? So, kind of neat. I just thought that was fun.
That's interesting.
Storing fatty acids. So fatty acids are great at storing energy because they have lots of carbon-carbon double bonds. If we think back to acetyl-CoA, one of the things that made it awesome is that we could use that to transfer two carbons. So we could build larger carbon structures like fatty acids. Okay? So we can use them to store. Unfortunately, just a bunch of single fatty acids floating around then provides too much, so we want to compact that even further. Okay? So hydrogen fuel cars aren't particularly efficient because we can't compact enough hydrogen fuel down into a small enough space to make them particularly useful. Okay? So we want more storage within each of these kind of processes. Plants have come up with ways to, and animals for that matter, have come up with ways to store the fatty acids to make that energy more compacted and more dense. That goes into making waxes in the case of plants. Okay? So in the case of a plant, what we've got is a massive fatty acid coming out one end, and at the other end of it, okay, we end up tacking on another big long carbon chain. Okay, and this does a couple things for us. Number one, it hides the fatty acid. Now we don't have a hydrophilic end and a hydrophobic end. Now the entire molecule is hydrophobic, but we've at least stored in all those carbons. By making the entire molecule hydrophobic, what have we then made? Don't tell me wax. We now have a protective coating. Okay? If you dump the wax on the outside of the plant, what happens? All the water that's in the plant stays in the plant. We get rid of that wax coating, what can happen? Particularly in the desert, the water can evaporate out. Okay? So plants have used this kind of fatty acid mechanism to help keep water present within the plant. Okay? Kind of a fun little deal. So it is used typically as a protective coating. We can go through and isolate waxes and convert them back into the fatty acids to make biodiesel. Okay? So lots of our biodiesel comes, or some of our biodiesel comes, from burning fatty acids. We have to get the fatty acid accessible and reactive before we can actually combust it for our biodiesel. So we have to break down the wax into our fatty acids. And once we're in the fatty acid, we can then process that a little bit cleaner or easier. The other process is going into triacyl glycerols. Okay. The triacyl is referring to this functional group. Whoops. That was the wrong eraser. This is known as an acyl group. Okay. If we look at our final molecule here, how many of those acyl groups do we have? 3. So we have a triacyl system. Okay. The glycerol part of the name comes from the part that's holding all 3 of those acyl groups together. And that molecule that I've got highlighted in purple is known as glycerol. So we have triacyl glycerols. So glycerol is just kind of our nice, simple, branched 3-carbon derivative of a... That's a harder question. What is glycerol a derivative of? No. There it is. It's a derivative of carbohydrate. It is the reduced form of glyceraldehyde. It is the reduced form of our dihydroxyacetone, okay, that we saw back in glycolysis. So if we reduce one of those molecules down, we can get a glycerol. The glycerol can then be used to dump a bunch of acetyl groups onto, or a bunch of carbons onto. Okay. Is there a question there, Mike?
I was just trying to think of the structure of diet.
I can draw it. So our dihydroxyacetone. So acetone is this guy with 3 hydrogens out at either end. The dihydroxy is because we replace one of those hydrogens on each side with an OH, and we now have our dihydroxyacetone, okay, which is related to our glyceraldehyde, there. Okay, so they are isomers of each other. If we reduce either of those, we would end up at a glycerol. Okay. And if you think even further back to our carbohydrates, we talked about the reduction of our carbohydrates. Sucrose, I think it was sucrose. When we reduce sucrose, we can reduce it to, I might be wrong on this, sorbitol. Okay. When we did the reduction down, we changed the name of our carbohydrate to an ol for the reduction. So glycerol is coming from glyceraldehyde. Okay. And when we reduce glyceraldehyde, we have glycerol. Okay, we've gotten rid of the carbonyl. So there's our fun Medusa looking like pictures for our triacyl glycerols. To go ahead and process these, you can use lipases. And what lipases will do is hydrolysis. So they will break the weakest bond within these structures, or within your triacyl glycerol. The weakest bond being the one I'm now highlighting in red, to reverse the reaction. So our lipase does that. Interestingly enough, back with the name, yeah, a lipid was our fatty acid. If we're now doing a lipase, we're doing an enzyme acting on a lipid. There it is. If we do the same reaction, but not using enzymes, we do it as humans, we call the reaction a saponification. Because what we end up producing is, yes, the glycerol, but then we also produce the fatty acid. And when we go through and produce the fatty acid, we end up doing it under basic conditions, and we form that structure, the one in our red box. And that structure is known as soap. Okay. So if you ever wanted to make soap at home, you can buy animal fat, add a bunch of base to it. Be careful with the amount of base you add, otherwise the soap kind of becomes dangerous. Okay. And let the base react with the soap, or with the fatty acid to break down the fatty acid into the soap molecules. The glycerol will help act as kind of a binding agent and help with lubrication when you use the soap. You let that dry out, you now have soap. Okay? So rubbing animal fat all over yourself doesn't do a good job of cleaning. You need to add the base to cleave it from the glycerol. And once you've got that negatively charged end on your fatty acid, you now have a soap, a very hydrophilic head group and a very hydrophobic tail group to help pick up dirt molecules. Okay? So yeah, in humans, that's saponification. So just in case anybody starts quoting me, human ACE I made up. Clarify. Because humans did it, and it's an enzyme. You gotta have fun somehow. Now we can go through the billions of different ways that we can modify our triacyl glycerols. Because we have those 3 alcohol functional groups, we can attach fatty acids to those, or we can attach other groups. Those other groups will change the physical properties of our acyl glycerol and can change what its function might be later on down the line. So in our first case, we can go a phosphoacyl glycerol. So all we end up doing is taking our glycerol and we put a phosphate on it. Okay? That phosphate is now charged, which then means we've changed how that molecule interacts with its environment. Okay? It's going to be much more water soluble at that one end of it. We can then link on a bunch of our fatty acids, and we end up with, oh wow, great, an entirely new lipid. We'll call these all our phosphoacyl glycerols. Okay? Because they're still our acyl glycerols, but we've added a phosphate. Okay? How is that going to change the properties? Well, we're less storage now, and we have more hydrophilic character at one end group. Depending on which fatty acid we put on, so in this case they put on a sterol group and a linoleal group, right? That's just referencing the type of carbon chain you have. Okay? Be it the sterol group where it is just a straight chain of carbons, a saturated carbon system, or the linoleal group where you have some unsaturation within it, some double bonds. Okay, so each of those carbohydrates would pick up a different name. I don't think anybody would be expected to know the name of those. Okay, so don't panic on that. Oh my gosh, look, fatty acid sphingolipids. So of course we have an entirely new thing. You take glycerol and you modify it at 2 ends. Okay, one end you add in a bunch of carbons, and then you have your 2 alcohols, and then your fatty acid connection goes in through the middle. What is this going to go through and change? Well, now you've lost a lot of the hydrophilic characteristics of this. Okay? It is still present, but it's not quite as big. Okay? And we only have 1 fatty acid connection on this. This changes the properties of our hydrophilic head group, which then changes where it can potentially interact and what it might do. Okay? Some of the names that come out of this: sphingosine is the glycerol part of the sphingolipid. So the sphingolipid is sphingosine with, I can already see people getting mad at me. I didn't create this. The sphingosine is your original glycerol molecule, or your modified glycerol molecule. So our glycerol, okay, is this kind of general backbone. Notice it's modified because we have a nitrogen in there, and it's also modified because we have this extra carbon chain up at the top. Okay? The fatty acid is still being connected in, it's just being connected in an odder fashion. Okay? So your sphingosine is the equivalent of glycerol. If we further change the sphingosine, okay, by adding a fatty acid to it, we now call it a ceramide. Why? Because biochemistry hates you. Right? If we then further mess with it, we look at the lower right-hand corner. If we take that alcohol, the last alcohol on our sphingosine, and we add a phosphate with, what looks to be a modified, actually I have no idea what that looks to be a modified of. A small carbon chain with a nitrogen on it. Okay. We now have sphingomyelin. Okay. So again, subtle changes to our glycerol molecule. We're still bringing in our fatty acids. These subtle changes change the properties of the lipid, which then means it's used in different locations. This becomes important because these are found primarily in the nervous system. Okay? Our nervous system, we need to kind of protect and isolate. We protect and isolate it by dumping a bunch of fat on the outside of it. Okay? Why do we want a bunch of fat on the outside of it? Our nervous system works by transferring electrical impulses. Okay? What is fat good at? Insulating it. So it kind of protects those electrical impulses within the nervous system so it doesn't branch out, number one, to cause electrical shock, which could be bad. And it also then allows for faster signals through your nervous system as opposed to them getting delayed and washed out into different directions. So it kind of keeps them condensed. Okay. Yay! More lipids. So we take our glycerol molecule, or actually our sphingosine, and we add, instead of just an OH to become a ceramide or that weird phosphate to become sphingomyelin, we add a carbohydrate at that position. Okay. So our sphingosine now has a carb attached there. As soon as we attach the carbohydrate, we now change the name of our lipid again. Okay. Which kind of makes sense because it is a different structure. Okay. And in this case, we refer to it as a glycolipid. Why might we refer to it as a glycolipid? The sugar that we attached was glucose. Okay? So we can refer to it as a glycolipid. So it is usually glucose or a galactose. If you add on a whole bunch of crazy carbohydrates, you can then refer to it as a ganglioside or ganglioside. No, I had it right. Ganglioside. Okay, which I think I've got on the next. No, I decided I was done listing them and showing pictures. So you use a crazy big carbohydrate mass, you can end up with gangliosides. Okay. Again, same idea, you're just putting on a bunch of different pieces, and this is how they then build these lipid systems. Okay. What can we do with these lipids? That's our biological membranes. Okay. Interesting features between animals and plants are animals use saturated fats. Those saturated fats stack really, really nicely against each other, which then means it doesn't move very well. Our membranes are not very fluid, which makes it a lot more difficult to pass things in and out of them. One of the things we do to kind of help that fluidity is to incorporate other molecules into it, like membrane proteins, which will be important in both cases, but cholesterol. Animals use cholesterol to help with membrane fluidity. How are they going to help? Let's see, do I have a picture? Oh, there it is. Cholesterol is that big ring structure. That then opens the spacing between our lipids, which allows for a little bit of motion, or a little bit more motion, and a little bit more fluidity. Okay? So that's why cholesterol helps us out with it. Okay? In our plants, they're unsaturated fatty acids, which then means we get more motion because those fatty acids then kind of break up the stacking interaction between all of them. Okay? Depending on which kind of system we're looking at how many fatty acids are involved, we end up with differing levels of kind of characteristics. You've got micelles, you're looking at a very small number of fatty acids generating a very small sphere, in which case you really only have a hydrophilic outer group and a hydrophobic intersection. If we make that sphere large enough, we can get a bilayer, okay, 2 setups, getting us a liposome. We refer to that one as a liposome because it's a relatively small interior hydrophilic group. Okay. If we make that even larger, that's when we move into our full bilayer sheet. Okay. When we look at cells, they aren't just flat sheets. They are round. So they are effectively a liposome. It's just a really, 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 really big liposome so that it is effectively seen as a flat surface, much like the planet.
Except when the center, it's,
Yep. So a cell is a massive liposome, is really what it comes down to. Okay. Membrane composition. So you'll notice that when we take a look at this, we're showing an inner and an outer membrane. Okay. Any interesting observations that you note? Nothing? You're like, there's no difference whatsoever
Well, no,
between them?
'cause since it's hydrophilic, there's charges. There's charges
Yeah.
all on the outside, and then the inside
True.
is hydrophobic.
I completely
no
agree.
charges.
So we're getting the exact same kind of charge buildup on either end. There's no other difference?
It tends to be more of the
So like all green in here being our phosphoacyls, our P1, whereas on the outside, okay, you're getting a whole bunch of different ones popping up. Okay, that's a bit weird. Why might that be the case? Let's go back
be
to
able to communicate with the outside.
something else. Glycolipids. What do we have?
The sugars.
Our head group has big or small? The hydrophilic end, big or small? It's really, really big. Sphingolipids, really, really big. Our phosphoacyl glycerols, relatively small. Okay, even our triacyl glycerols, relatively small. Okay, why is that important when we look at the membrane composition?
Because it's round. You need it.
The inside is going to be compressed a little bit smaller, which means the head groups need to be smaller because they're getting closer to each other. If they were really big, we can't get enough groups within there, and we end up losing some of the integrity of our membrane. Okay? So the outside of our membrane tends to have a lot more of the larger head groups because there's more space on the outside. Okay? So we can make that a little bit more porous, potentially, and the inside can be a little bit tighter packed. Okay? And I have to stop there, though I thought I was going to get all the way through lipids.